Light is a mysterious thing that has puzzled scientists for many years. As Johnson described it long times ago, we all know what light is, but it is hard to tell what it is. Light is a mysterious thing that separates day from night. Nothing matches the speed of light, that is 300,000 kilometers per second. To our eyes, it appears as white, but actually it comes in many colors like violet, indigo, blue, green, yellow, orange and red, collectively appearing as white. The natural sources of light are sun, moon, lightning, certain insects and fish. The artificial sources of lights are lamps, torches, candles, etc. Light emitted from sun is a form of electromagnetic radiation. Radiation is energy that is transferred in the form of electromagnetic waves. When we talk about biological systems, light satisfied two most important functions. To power the energy systems or biological systems, that is to provide energy, and it also gives us important information about our environment or to trigger different events. So the energy derived from sun helps to maintain the thermal balance of our planet. That is our planet's surface temperatures, and sunlight drives the most important plant process on earth. That is the subject of our discussion, the process of photosynthesis. Besides providing energy for maintenance of or sustenance of different biological functions, sunlight also gives important information to trigger different developmental events in plants. For example, the initiation of flowering, certain plant movements, chloroplasts can adjust their position under fluctuating light conditions along with cyclosis or cytoplasmic streaming. The light energy also gives important clues for stomatal regulation, for opening, and it helps us in measuring day length. That is crucial for the initiation of flowering and some other events in plants' life cycle. So these different phenomena like photosynthesis, photomorphogenesis, that is the development of form and structure under the influence of light, photoperiodism, they are studied in an important discipline what we call as photobiology. Light is a form of electromagnetic radiation emitted by the sun. The electromagnetic radiation is a large family consisting of 7 members, which are gamma rays, X-rays, ultraviolet, visible light, infrared, microwaves and radio waves. The visible light is a narrow band of energy within this continuous electromagnetic spectrum that has a wavelength of 380 to 770 nanometer. These different types of radiation can be differentiated based on their wavelength, frequency and their energy content. A major discovery in the late 19th or early 20th century was the discovery of dual nature of light. That light is both a wave and a particle. So let us first discuss the wave properties of light. Imagine a wave in an ocean that is different from a normal elevation. How is it different? Because it has successive crusts and troughs. We can measure the wave-like properties by measuring the wavelength or frequency. Wavelength is the distance between two successive crusts or troughs, and we denote it by the Greek word lambda. So lambda is the distance between two successive crests or troughs. And we can also measure frequency. Frequency is denoted by nu, that is number of waves or cycles passing a point in space per second. And we can also write its formula here, nu is equal to c over lambda, where c is the speed of light, that is 3 into 10 raised to the power 8 meter per second. So we can measure the wavelength and the frequency. Usually the wavelength in biological system is measured in units of nanometer. 1 nanometer is equal to 10 raised to the power minus 9 meter. As light is a form of electromagnetic radiation, so it has two types of waves or fields, electric and magnetic. These electric waves and magnetic waves oscillate at 90 degrees to each other and to the direction of propagation. Light behaves like a particle when it interacts with matter. Light is also a particle which we call as photon. And each photon contains an amount of energy which we call as quantum. It's plural is quanta. According to Planck's law, we can write down the equation for energy content of a photon, that is h nu, where h is Planck's constant with a value of 6.626 per photon. So this is the value of Planck's constant. So we can also write this equation as mu is equal to c over lambda. We can also write this equation as hc over lambda. So you see that the energy content of a photon is inversely proportional to its wavelength. So shorter the wavelength, more the energy, and longer the wavelength, less is the energy. Imagine a calm sea and a turbulent sea. In a turbulent sea, you will see waves going like this, while in a calm sea, you will see waves going more like this. You also see this in a picture showing the electromagnetic radiation spectrum. So shorter waves like gamma rays or X-rays are like this, whereas the longer waves like microwaves or radio waves are going like this, slower and slower. So shorter waves have more energy. That is why gamma rays or X-rays have too high energy content, and usually they are dangerous. Whereas these radio waves, they have less energy content, but they travel over long distances. Light affects both the growth and development of plants. Growth is irreversible, permanent increase in dry weight of plants. So this is a result of the process of photosynthesis mainly. Whereas development is an umbrella term, here we consider development as changes in form and structure of plants. So we can call it as changes in form and structure of a plant, or we can describe it as changes in shape. The principal difference between growth and development is that growth is determined by the amount of radiation, whereas development is determined mainly by temperature. For example, if we are growing rice in a growth chamber under controlled environment conditions, so for proper growth of rice, we may need 600 to 800 micromole per square meter per second of light intensity in the growth chamber, whereas for developmental response only 10 to 15 micromole per meter square per second is enough. So light affects both these processes, growth as well as development, but growth and development are two independent processes, we can say. So while discussing the effects of light on plants, we must segregate the growth and development, whether light is affecting growth or is it affecting development, that in turn may affect growth. In photobiology, we are interested in measuring three different aspects of light. First is light quality. Light quality is the composition of light with respect to its wavelength, what we perceive as different colors like violet, indigo, blue, green, yellow, orange or red. And sunlight appears as white. So this is also called as spectral composition or spectral energy distribution. So light quality means which wavelength of light. The second important aspect is light quantity. Light quantity means how much light a plant has received. So this is measured in terms of light intensity. And this primarily affects the rate of photosynthesis or net photosynthesis. And third factor is light duration. That is the number of light hours in a day. So this depends on equator and also depends on other factors like cloudiness or the level of air pollution or angle of sun. As we mentioned that light affects both growth and development of plants. So different wavelengths of light have different effects on plants. We use the term photosynthetically active radiation what we abbreviate as PAR. PAR is the range of wavelengths that drive the process of photosynthesis, as the name implies, photosynthetically active radiation. So we considered 400 nanometer to 700 nanometer wavelengths as photosynthetically active radiation. So these wavelengths are most effective in driving the process of photosynthesis. Why is this range most effective in driving the process of photosynthesis and not the other wavelengths? For example, on this side of graphs we have shorter wavelengths with high energy, and here we have longer wavelengths with low energy. Actually, the plant pigments can absorb this wavelength, and this wavelength is just the right amount to allow the flow of electrons. If we supply plant with low wavelength, shorter wavelength radiation, but with too much energy, too high energy, then this can knock the electrons off the orbitals, or they can damage the pigments. And if we supply plants with longer wavelengths but with much shorter energy, they have too low amount of energy to excite an electron. Like in microwaves, we can warm the meal. So similarly, if we provide longer wavelengths to plant, this can warm the pigments like chlorophyll, but they will not actually excite electrons. So this wavelength, 400 nanometer to 700 nanometer, is just the right wavelength for driving the process of photosynthesis. And usually we measure this wavelength, or we consider this wavelength when describing different plant responses. So in terms of photosynthesis, this is true. But actually there are other wavelengths that are also important in different biological processes. So we use another term that is called as non-biologically active radiation. And this ranges from 300 nanometer to 800 nanometer. A main limitation of measuring PAR is that PAR sensors only measure wavelengths between 400 and 700 nanometers. But actually there are other wavelengths, for example, far red light above 700 nanometer, that is roughly 700 to 750 nanometer, that have other important effects in regards to photomorphogenesis. So light quality or wavelength of light affects different growth and developmental responses. For example, UV light or ultraviolet light is important for pigment synthesis in plants, such as the synthesis of anthocyanins. Blue light is important for pigment synthesis and is also important in stomatal opening. And blue light inhibits cell expansion. So it makes plants shorter or more compact. Then we have red light that has important role to play in cell expansion or elongation, as well as for initiation of flowering cycles. Similarly, far red light also promotes cell expansion and makes plants taller. Recent advancement in controlled environment production is the use of LEDs, LED lights or light emitting diodes. Based on the research of 70s, researchers believe that only blue and red light are most effective in driving the process of photosynthesis. And LEDs with blue and red wavelengths were installed in greenhouses. But they did not give the desirable results. They did not give the highest yields. So now we know that the actually the entire range of spectrum, the portion of spectrum between 400 and 700 nanometer is effective in driving the process of photosynthesis, and there are wavelengths above 700 nanometer like far red light that has effects on plant shape or plant development. Similarly, red to far red ratio is important for different developmental responses like initiation of flowering cycles. So there are two terms. Absorption spectrum shows that which wavelengths are most strongly absorbed by a pigment, whereas action spectrum shows that which wavelengths are most effective in driving or powering a photochemical process like photosynthesis. We see that chlorophyll is absorbing blue and red light. So we see two peaks in the regions of blue and red lights. But if we look at the entire range of wavelengths between 400 and 700 nanometer are participating in the process of photosynthesis. We see a peak in red followed by blue and some green light. So that is why if we superimpose absorption spectrum over action spectrum, you see that the entire range of wavelengths is effective in driving the process of photosynthesis. So how does this happen? Because there are some accessory pigments like carotenoids which absorb in the regions of greens and yellow, and they can penetrate deeper into the canopies. When in canopies the blue and red photons are absorbed at the top, the greens and yellows can penetrate deeper into the canopy and help photosynthesis in the shaded parts, in more shaded parts. So this is the difference between action spectrum and absorption spectrum. So based on research on the response of plants to different colors of lights or different wavelengths of light, we can say that we need to prepare LEDs when we are optimizing the controlled environment production, which have different ratios of different wavelengths of light. So now the horticulturists are interested in lights, LED lights that have a particular ratio of blue as well as far red light. And this also depends on species. For example, in lettuce, the far red lights causes rapid expansion of leaves horizontally, whereas in tomato it makes plants taller. So more research on different wavelengths of light could possibly revise our definition of photosynthetically active radiation or power. Second important aspect is light quantity. That how much light a plant has received. We can quantify this by measuring light intensity. So when we are measuring light intensity, normally we can measure it as the amount of energy or radiation in units of watts per meter square, or we can measure it as number of photons or quanta, because it is the number of photons that drive the process of photosynthesis, not the total amount of energy. So the unit for this is micromole of photons per square meter square of green leaf area or any photosynthetic area per unit time or per second, and this is called as photosynthetic photon flux density. So measure photosynthetic photon flux density to quantify the amount of light that a plant has received. So this is micromole of photons per meter squared of any photosynthetic area like green leaf area per unit time or per second. There were other terms like lumen or lux, but they are not frequently used in scientific literature because they show the intensity of light at the source and not what the plant has received. There is another concept of daily light integral. Daily light integral or DLI. The concept of daily light integral is comparable to the concept of rain gauge, which measures the amount of rain on a particular day. So DLI is the summation of photosynthetic photon flux density. So this is the units for ppf or DLI are micromole per meter square per day instead of per second. So DLI is important indicator of how much light a plant has received in one day. So two types of sensors are used to measure light intensity. Depending on the geometry of plant or plant part. So if it is a flat surface like a leaf area, then we use the term irradiance. That is the energy flux per unit of flat surface. Whereas under practical situations, light is coming from different directions. For example, if this is a plant, so light is coming from different directions, and also if it is a sandy soil, this is also reflecting back to the plant. So in this case, we use another term, that is fluence. That is the radiant energy falling on a sphere divided by cross section of the sphere. So depending on whether we measure the number of photons, we can call as photon fluence, or if we measure the amount of energy, we can call it as energy fluence. If we look at the response of a single leaf to the light intensity, so we mentioned the unit micromole of photons per square meter of leaf area per second of power. We are not considering total solar radiation, because the total solar radiation cannot drive the process of photosynthesis. Only a fraction of total solar radiation, that is power, can drive the process of photosynthesis. And here we have net photosynthesis rate. So the response of a single leaf in different studies was like this. The light intensity at which the rate of photosynthesis is equal to the rate of respiration, or the carbohydrates that are being produced by the plants are being used by the plant in the process of respiration. So there is no net photosynthesis, or net photosynthesis is zero. That light intensity is called as light compensation point. Light intensity at which the rate of photosynthesis is equal to the rate of respiration. So there is no net storage in plant. As the light intensity increases, the rate of photosynthesis increases, and the total photosynthesis increases or exceeds respiration, then the net photosynthesis increases. But there is another point when further increase in light intensity does not increase net photosynthesis. This point is called as light saturation point, at which further increase in light intensity does not increase the rate of photosynthesis or net photosynthesis. So here there might be some other limiting factors like carbon dioxide concentration, response of photosynthesis for a single leaf. But if we look at canopy level, this response is more or less linear. That's almost linear at canopy level. Why is it so? Because in a plant canopy, the upper leaves, for example, imagine a forest in which the upper leaves are receiving enough light, but there are leaves that are shaded by the upper leaves and they are not receiving enough light deeper into the canopies. And the upper leaves might be light saturated, or they reach the light saturation point, but the lower leaves are still in the lower part of the curve. They are still light limited. So the response of a canopy to light intensity or micromole per square meter per second of power is linear. Third important aspect of light is light duration, that is the number of sunlight hours. This depends on latitude. For example, at equator the days are 12 hours long, and as we move to the south or north pole, the days are longer. In the middle latitudes, in winter, days are shorter. So this also affects the rate of photosynthesis. For example, if days are shorter, then the plants are able to accumulate low quantities of light and the rate of photosynthesis is low. But because the temperature is low and the rate of respiration is also low, they can still survive on the stored materials or stored nutrients in them. On a clear sunny day, the rate of photosynthesis is more than the rate of respiration. But in summer days, the amount of starch accumulated in the leaves can feed back, and in the negative feedback it can stop photosynthesis. The other important aspect, mechanism or plant response that is affected by light duration is well-known phenomena of photoperiodism, that is plant responses to the relative length of day and night. So according to the duration of light there and plant responses, plants can be short day or they can be long day, but there are some other plants which are day neutral, and within a species there is huge variation. Some varieties may be photoperiod sensitive while others are not.
High energy electrons are moving through these power lines, which could easily kill a person or anything that comes in contact with them. Photosynthesis also involves the movement of electrons, which, if not controlled properly, could damage the chloroplast and the whole plant. When light strikes photosynthetic pigments, an essential but risky process begins.
The flow of electrons in the so-called electron transport chain requires the absorption of light energy, and there must be something to absorb this light energy, what we call as pigment. Pigment is any material that absorbs certain wavelengths and therefore has a distinctive color. So photosynthetic pigments absorb wavelengths specifically, reflecting the others. For example, chlorophyll absorbs the blue and red wavelengths, blue photons and red photons, while refracting others. So what are the characteristics of these pigments? Pigments have alternating system of single and double bonds between carbon atoms, which give them their strong light absorbing properties. They can be excited easily and they can act as electron donors. So what would be an ideal pigment theoretically? An ideal pigment should be black, absorbing all wavelengths of light coming from the electromagnetic radiation spectrum. But we see that chlorophyll is green in color, and plants can use the wavelengths between 400 and 700 nanometer, what we call a photosynthetically active radiation. This is so because if a pigment absorbs wavelength of higher energy or lower wavelength, then those high energy radiation would knock the electrons off the orbitals of pigment molecules, and their whole structure would become useless. They would not be able to effectively carry out the process of photosynthesis. And if they absorb longer wavelengths, then that cannot be used to excite electrons. They can warm the pigments, but they cannot be used in photochemical processes like photosynthesis. So photosynthetic pigments absorb just the right amount of energy between 400 and 700 nanometers. So if a light strikes a photosynthetic pigments and if it has a wrong wavelength, for example short wave radiation with high energy content, it will be transmitted through the bonding orbitals without being absorbed. But if a right wavelength light in the photosynthetic range is absorbed by photosynthetic pigments, then the electron will move to the high energy orbital. So the lowest energy state of a pigment is called as ground state. So now this has entered the excited state. Its energy level is raised, which corresponds to energy level of this distant orbital. What happens when a pigment goes from ground state to an excited state? So here the principle of the first law of thermodynamics comes in action. The first law of thermodynamics is that the energy must be conserved. So when a pigment, for example chlorophyll, absorbs energy which is equal to h nu, then it enters or it goes to the excited state, which we note with this star or hashtag. So in the excited state, the pigment is less stable. The higher the excited state, the lesser is the stability. So it must return to its ground state. So how does this happen? There are different ways in which this energy is conserved. So suppose this pigment absorbs blue photon and it goes to a higher energy state. By absorbing this blue photon, this is in the excited state. So there are different phases. It can lose some of its energy as heat through molecular vibration and enter a lower excited state. So this is the lower excited state. This is the higher excited state and this is the lower excited state. Because blue photon has less wavelength and higher energy content, whereas red photon has longer wavelength and lesser energy content as compared to blue photon. So this is the process of absorption when it enters the excited state, and another process is what we call as fluorescence. We said that chlorophyll can lose its energy, some of its energy as heat, or the excited state, the molecule can lose energy by emitting light and regenerating a photon. This process is reverse of absorption, what we call as fluorescence. Or the molecules can transfer its energy of excited state to another molecule. This process is called as energy transfer. And fourth fate is use of this light in photochemical processes, what we call as photochemistry. this is the most useful process. So the lower excited state can be used in redox reactions of photosynthesis. What drives the process of photosynthesis? So there are two types of pigments. Primary pigments is chlorophyll A, which has been unaltered for about 3 billion years. And this is the principal pigment in higher plants as well as algae and cyanobacteria, whereas accessory pigments like carotenoids and chlorophyll B in higher plants, they broaden the absorption spectrum of photosynthesis, absorbing wavelengths which are not absorbed by chlorophyll A, and then once it is absorbed within the photosynthetic range, then green photons and blue photons and red photons are equal, because according to Stark-Einstein law, one photon can excite one electron. Photosynthetic pigments are amphipathic, meaning they have both hydrophilic and hydrophobic parts. Chlorophyll A and chlorophyll B are large flat molecules with a tadpole-like structure. They have a head and a tail. The head consists of identical porphyrin ring structures and which are hydrophilic, whereas phytotails are hydrophobic and dissolve into the lipid portion of the thylakoid membranes. When packed tightly in a membrane by their phytotails, their porphyrin rings or head-like structure lie parallel to each other. This causes the electron orbitals of one molecule to interact with those of the other two adjacent molecules. Hundreds of chlorophylls act somewhat like one molecule, and the energy absorbed by one can be rapidly transferred to another in a different part of the complex. This transfer is called as resonance, which allows chlorophyll B to absorb wavelengths that chlorophyll A would miss, and then to transfer the energy to chlorophyll A for use in chemical reactions. Carotenoids, however, are poor at this type of resonance and can only transfer approximately 10% of their energy. So their role is in absorbing excessive light and dissipating the excess heat, and thus protecting chlorophylls from photooxidation. Rather than occurring at random, the pigments are held in place by proteins called light harvesting complex proteins that are also embedded in the thylakoid membrane. The entire set of 300 chlorophylls, carotenoids and their proteins are called as antenna complex. When light strikes any pigment of an antenna complex, the energy is transferred to a reaction center. Reaction center is a structure that contains a pair of special molecules of chlorophyll A whose properties differ from rest of chlorophyll A in the complex. The reaction center's pair of chlorophyll A molecules are the only ones that can actually donate an excited electron onto an acceptor molecule. It is important to transfer or pass energy from one chlorophyll to another, because the particular chlorophyll molecule absorbs only a few quanta per second. So this is too slow to keep the later steps of photosynthesis operating properly. So by having one antenna complex, the several hundred pigment molecules all transferring their energy to one reaction center, which thus acts as if it is absorbing thousands of quanta each second. How chlorophyll functions in photosynthesis? There are two key experiments, quantum yield experiments by Emerson and Arnold, and the Emerson enhancement effect experiment, which are key to understanding the functioning of chlorophyll in the process of photosynthesis. In 1932,
Emerson and Arnold performed a key experiment that gave very useful insights into the details of photosynthesis. They found that chlorophylls don't work independently during photosynthesis. They supplied a green algae called chlorella with short pulses or flashes of light about 10 raised to power minus 5 seconds, and found that 9 to 10 photons were required to evolve one molecule of oxygen. Here in this graph you see photon flux density on x-axis that shows the number of photons falling per unit area per unit time, and on y-axis you see moles of oxygen evolved or the number of oxygen evolved. And you know from the equation of photosynthesis that the number of carbon dioxide fixed and the number of oxygen molecules evolved is equal. So for measuring the rate of photosynthesis we can either measure the number of carbon dioxide fixed or the number of oxygen evolved. So they found that 9 to 10 photons were required to evolve one molecule of oxygen. Before that it was believed that only one photon is required to evolve one molecule of oxygen, and they were able to saturate by providing the high intensity flashes of light. And they also found that to evolve one molecule of oxygen 2500 chlorophyll molecules are required. So this was the first indication that chlorophylls don't work independently in photosynthesis. They work in units together to facilitate these reactions. The main conclusion of this experiment was that there are many photons and many chlorophylls required for evolving one mole of oxygen. In another key experiment, Emerson used different colors of light. So using red and far red light, he eliminated algal culture, first with low intensity far red light and by observing oxygen evolution, then eliminating the algal culture with red light and observing the oxygen evolution. Then he turned on both red and far red, and it was amazing to see that the combining of red and far red light gave an effect that was 40% greater than simply a sum of the effects of red and far red light on oxygen evolution. That is why this is called as the Emerson's enhancement effect. So something happened that enhanced relative rate of photosynthesis when both red and far red light were combined. So the conclusion was that chlorophyll molecules are not homogeneous. There are two photosystems. So this was the first suggestion of two photosystems, photosystem 2 and photosystem 1, which work in series.
We had talked about different aspects of light, the characteristics of photosynthetic pigments, and now in the last part of this lecture I'll talk about the electron transport chain. Let me draw the Z scheme of electron transport chain here. First, the photosystem 1, whose reaction center chlorophyll absorbs preferentially 680 nanometer of light and goes to a higher energy state. So here we have the electron donors, and here we have good electron acceptors. So it cannot remain in this excited state for long. This lasts for only 10 raised to power minus 14 seconds. But now this has become a very good electron donor, so it can donate its electron to an electron acceptor. So now there is a series of electron acceptors in this chain. First, this will transfer its electron to few Photon, which will transfer its electron to plastoquinones, converting it to PQH2. PQH2 is oxidized at the cytochrome B6F complex, which transfers the electron to plastocyanin, a copper containing protein, which is a mobile. Plastoquinones and plastocyanins are mobile carriers. Plastocyanin delivers the electron to photosystem 1 that absorbs preferentially at 700 nanometer. So when photosystem 1 absorbs light energy, it goes into the higher energy state. We denote this like this. This is also a good electron donor. Now it can donate its electron to first to A0, then to ions, sulfur complexes. The exact nature is not clear yet, which can transfer electron to a small protein ferredoxin. Ferredoxin transfers it to ferredoxin NADP reductase enzymes. And this NADP reductase enzyme can reduce NADP to NADPH. This is how the reducing power NADH is formed in the so-called electric transport chain or Z scheme. So that is how NADPH is formed. So on this side we have the electron acceptors, and on this side we have very good electron donors. And the whole purpose of light is to produce good electron donors and good electron acceptors. So, and we need energy input at 2 points, at photosystem 2 and at photosystem 1. The rest of process is downhill and does not require energy. So the source of electron in photosystem 1 is actually the photosystem 2, because photosystem 2 on absorbing light initiates reactions that ultimately deliver electrons to photosystem 1. So now photosystem 2 has positive charge on it, and it is deficient in electrons. It is hungry for electrons. So where does this replenish its electrons from? It replenishes its electrons by splitting off water molecules. So it splits water molecules into oxygen, protons and electrons, and which can be then again carried through different electron carriers to photosystem 1. This is called as oxygen evolving complex or manganese. And when PQH2 is oxidized at cytochrome B6F complex, it pumps protons from stroma into lumen. Now there are two sources of protons. By pumping of protons from stroma to lumen by plastoquinone and the splitting of water molecules. The lumen is impermeable to the transport of protons. The movement of protons from higher concentration in the lumen to the lower concentration in the stroma is facilitated by a mushroom-like structure called as ATP synthase. What we call as the proton motive force or the electrochemical potential gradient according to the Mitchell's kinematic hypothesis. The energy of these protons is used to force a phosphate group onto adenosine diphosphate, thus converting it to ATP. That is how the ATP or adenosine triphosphate, which is the energy carrier. So while a plant produces ATP and NADPH, instead of using the light energy absorbed by pigments directly into the metabolic reactions. There are two reasons. One is that pigments are large molecules. They are not very mobile. They cannot move across membranes. Second, they absorb a huge amount of energy and therefore they cannot participate in all metabolic reactions. They will do more damage than the benefit. So the energy absorbed by those pigments is then converted into small packets of energy, what we call as adenosine triphosphate or ATP. This ATP has 3 phosphate group, and when one of its phosphate groups splits off and it is converted into ATP, the high energy is that is just right for forcing almost any of the metabolic reactions to proceed. The second phosphate group is rarely split off. Usually it is one phosphate that is split off. And these ATP molecules are charged batteries. These are like small charged batteries, make only a tiny fraction of the plant body, and they can be recycled and reused many times in the metabolic reactions. So that is why the light energy is stored as chemical energy in the form of ATP and NADPH. Along with these energy carriers, ATP plants also need the reducing power that is produced in the form of NADPH.
Our atmosphere has about 21% oxygen. So we have an oxygen rich environment due to which many compounds are found in their oxidized form. For example, carbon as carbon dioxide, sulfur as sulfate, and nitrogen as nitrate. Oxidized means that an atom does not carry as many electrons as it could. However, within the organisms, unlike our environment, many compounds are in the reduced state. For example, carbon is often in the form of carbohydrate, and sulfur is present as SH2, nitrogen is present as amino groups. So thus, in addition to needing energy, organisms also need reducing power, that is the ability to force electrons onto compounds. Reducing power is important for plants because they take in carbon dioxide and water, which are most highly oxidized form of carbon and hydrogen, and then convert them to carbohydrates, fats, and other compounds that are very reduced. When NADPH reduces a compound by transferring its electrons to it, the proton is released and NADP is regenerated. So rather than having a large number of carrier molecules, the cell recycles each molecule using it thousands of times a second as it shutters between electron producing and electron consuming reactions. As in case of energy, the optimum solution for moving and handling this reducing power or electrons is to use small molecules that are semi-stable and mobile. Therefore, ATP and nicotinamide adenine dinucleotide phosphate, ATP and NADPH, uh, are energy carriers which are ideal for transfer of electrons and for supplying energy for any metabolic reactions. So the summary of the second part of the lecture or electron transport chain is that in higher plants the major mode of electron transport is non-cyclic or linear electron transfer, in which water is oxidized to oxygen and NADP is reduced to NADPH. This requires photosystem 1 and photosystem 2 to work in close collaboration. Photosystem 2 preferentially absorbs red light of 680 nanometer and oxidizes water to molecular oxygen in the thylakoid lumen. In this process, it releases protons in the lumen and transfers electrons to plastoquinone to ultimately form PQH2. This mobile carrier then moves to the cytochrome B6 F complex where PQH2 is oxidized. From cytochrome B6 F complex, electrons move to the plastocyanin, the copper protein carrier, and oxidation of PQH2 in the cytochrome B6 F complex is also coupled to proton transfer to the lumen, which adds to the proton motive force. Plastocyanin moves to photosystem 1 where it reduces oxidized for P700 or photosystem 1. Photosystem 1 preferentially absorbs light at 700 nanometer and facilitates electron transfer from NADPH, NADP to NADPH in the stroma by the action of ferredoxin and ferredoxin NADP reductase. ATP synthase uses the proton gradient to produce ATP or adenosine triphosphate. Adenosine triphosphate and NADPH are then consumed in the carbon fixation reactions within the stroma. Plants are also capable of alternative electron transport process, including the cyclic electron flow. In cyclic electron flow, electrons flow around photosystem 1 that results in ATP synthesis but does not involve a terminal electron acceptor. So electrons are passed from ferredoxin to plastoquinone, thus increasing the pH gradient. This process adjusts the ATP and NADP production ratio, and this is important when linear electron flow is inhibited, for example, under fluctuating light conditions. So the main difference between cyclic and non-cyclic electron transport chain is that in cyclic only photosystem 1 is functional, whereas in non-cyclic electron transport chain both photosystem 1 and photosystem 2 are functional. And NADPH2 and oxygen is formed in non-cyclic electron transport chain, which are not formed in cyclic electron transport flow. The protons that drive the ATP synthase motors, they are not consumed themselves in this process, but their concentration gradient allows the forcing of phosphate group onto ADP, thus converting it to ATP. In subsequent lectures, we will discuss how ATP and NADPH, the energy carriers, are used to fix carbon dioxide in carbon fixation reactions or the so-called dark reactions.